now we're going to see the anatomy of the eyeball. We have three layers: the external, internal, and medial layer. The medial layer is the vascular layer. The external layer is composed by the cornea and the sclera. I will put them as we go by. So this would be the sclera, and here would be the cornea, more or less. The internal layer is just the retina, and the vascular layer is the uvea. Here we have posterior uvea and anterior uvea. The posterior uvea is the choroid, and the anterior uvea is the ciliary body and the iris. So all these are continuations. So here we have the choroid, and here is continuous then with the ciliary body. Then the iris. Here the iris. Here we have the pupil. The pupil is a hole. It's just the hole between the iris. It's not a structure, so what we are seeing would be the inner layer. The inner layer would be the retina. Here is the retina. Oops. Alright, so here the iris have this arrangement. Well, this is the iris. Well, without this part. This is the iris. Remember, if we see the eye like this, we have the pupil and the iris that gives the color of the eye. Could be green, could be brown, could be blue. Alright, so the external layer is the cornea and the sclera, and all the sclera is covered by the conjunctiva. The cornea does not. So we have a posterior chamber and anterior chamber. The posterior chamber would include the lenses that are suspended by this sonula fiber of the ciliary body. So this is the ciliary body, this is the iris, this is the lens. So the posterior chamber is all this. Goes from the actually the anterior, let's say, wall of the posterior chamber is the posterior wall of the ciliary body and the iris, and the anterior chamber, this is the anterior chamber. The posterior wall of the anterior chamber is the anterior wall of the iris and the ciliary body. The anterior chamber has aqueous humor, and this posterior chamber has the lenses and the vitreal cavity. The vitreal cavity has vitreous humor that contains water and proteins, proteoglycans, collagen, etc. So this is more like a gel. Alright, so the production of the aqueous humor is by the ciliary process, and it is sent here to the posterior chamber, and then through the pupil, here is the pupil, will pass to the anterior chamber and will be drained here at the Schlemm canal. The Schlemm canal is on the sclera and is very near to this angle. This angle is the iridocorneal angle, or the angle of the anterior chamber, and is when the iris inserts onto the kind of the sclera and the cornea in the junction between those two. So when there is glaucoma of close angle glaucoma, is because this angle is narrow. Alright, now let's see specifically each part. The lenses has a capsule that cover all. There are fibers with a nucleus, and we have the epithelium, subcapsular epithelium. Alright, now let's focus on the external layer first. We have the sclera. The sclera is continuous with the dura mater of the optic nerve. Here's the optic nerve and here's the dura mater. The sclera has layers. One of it is the fossa layer. This layer will penetrate the choroid at the space, perichoroidal space. Also, the sclera has two kinds of blood vessels, the superficial and the deep ones. So, when we see hyperemia, it's because there are dilation of the deep vessels. Now, why we know that there are deep vessels? Because if we give the patient phenylephrine, we will see that the superficial vessels constrict and we will only see these ones, for example in uveitis. But also the conjunctiva that covers the sclera. Here we have the eyelid, here we have the eyelid, here we have the eyelid. Then we have the white part, that is the sclera, is this part, the sclera, and then only on the pupilla we have the cornea. You see the cornea, I put it very big. More like this. Alright, so on the junction between the sclera and the cornea, this is the sclerocorneal limbus, and there are the regenerative cells of the cornea. Also the junction of anastomosis between the superficial and deep vessels of the sclera, and also between the blood vessels of the sclera and the conjunctiva, because also the blood vessels of the conjunctiva, we will see hyperemia in conjunctivitis, etc. So when we see hyperemia of the eye, it's because there are vasodilation of the blood vessels of the conjunctiva and of the sclera, mainly the deep vessels. Remember, the cornea has no vasculature, it's avascular. There are layers on the cornea that include the Descemet membrane and the Bowman membranes. These are the anterior and posterior laminas. Alright, so in the cornea we have a layer of cells that has microvilli, and so they trap the mucus produced by glands of the conjunctiva, and so the cornea has the humidity. Even though they dehydrate the cornea, they contain the tears. Alright, now on the anterior chamber we have aqueous humor. Now let's see the iris, ciliary body, and choroid. So the choroid here has pigmented, a little of pigment, epithelium, or just some pigment, because there are a few melanocytes, there are some macrophages. We already saw that at the perichoroid space there is penetration by the sclera. The choroid goes from the end of the retina, that is here, the os ora serrata, to the beginning of the optic nerve. The choroid has the external, medial, and internal layers. All of this contains blood vessels. The external layer is going to supply the sclera, the internal will supply the retina, and the medial would be continuous with the ciliary body and the iris. Now, we have the these blood vessels, they have largest caliber on the inner layer, and this is called the choroidocapillary layer. And beneath this we have the brush membrane. The brush membrane is in contact with the retina, with the pigmented epithelium of the retina that we will see on a while. Alright, so this brush membrane, actually we have the brush membrane has 5 layers. The endothelium, that is the endothelium of this choroidocapillary layer, then a collagenous layer, a elastic layer, another collagenous layer, and the lamina of the pigmented epithelium of the retina. Alright, so now the ciliary body and the iris would have pigmented and non-pigmented epithelium. We will see them in specifically. That would be continuous with the retina, not the well, yes, with a little of the pigment of the choroid, but most of the time with the retina. So now let's go to the ciliary body. The ciliary body contains the ciliary muscle and the ciliary processes. This is the ciliary muscle, and these are the ciliary processes that are deriving the sonular fibers that they make the suspensory ligament of the lenses. Here also the ciliary process are on the pars plicaris. The sections of the lens are the pars plicaris and pars plana. The pars plana is just the access to the vitreous cavity, and the pars plicaris is where the ciliary process are, and where we have the deep or superficial inner layer and superficial layer. The superficial layer is where we have the ciliary processes, as well as the production of the aqueous humor and the pigmented epithelium of the ciliary body. And on the inner layer we have the non-pigmented epithelium. And the muscle, here is the muscle. The muscle is innervated by the parasympathetic pathway in which the oculomotor nerve is involved, and when there is innervation of the ciliary body, there will be contraction of the ciliary body, decreasing the tension of the sonular fibers, and so the lens will have a more round shape, and we will be able to see near objects. Now let's go to the iris. The iris has a posterior epithelium and a anterior myoepithelial cells. So the posterior epithelium is the posterior pigmented epithelium and is the main pigment of the iris, and this would be continuous with the pigmented epithelium of the ciliary body, and then would be continuous with the pigmented epithelium of the retina. The anterior myoepithelial cells, we have the basal cells or the anterior cells and the posterior cells or apical cells. The apical cells have pigment, so this would be the anterior pigmented epithelium, and we have these basal cells that projections of these would give the dilator of the pupil and sphincter constriction muscle. So here we have the pupil, around we have the constriction of the pupilla, and radially we have this dilator muscle. So when there's contraction of the dilator muscle, there would be mydriasis. The constriction of the sphincter muscle, there would be meiosis. So the anterior pigmented epithelium has less pigment, but contains a little of pigment that most of the time is covered by that of the posterior pigmented epithelium. Now, the anterior pigmented epithelium will be continued with the non-pigmented epithelium of the ciliary body and with the neuroretina. Alright, so we already saw that the iris gives color to the eye and functions like as a diaphragm. So depending on how mydriasis or meiosis we have, is the size of the pupil. The pupil is just a hole, and what we see through the pupil is the retina. Now let's go to the inner layer, that is the retina. The retina has 10 layers. The pigmented epithelium of the retina, the photoreceptor layers, that is where the cones and rods lie, the outer limiting membrane, that is where the outer limitation of the Muller cells is, then the external nuclear, this is where the nucleus of the photoreceptors lie, external plexiform, that is where the interaction or the synapses between the photoreceptors and the bipolar cells occur, and also some interaction between other cells like amacrine cells, horizontal cells, etc. And then again we have now a internal nuclear, that is where the body of the bipolar cells, amacrine cells, horizontal cells, etc. are. Then the internal plexiform, that is where the synapses between the ganglionic cells and the bipolar cells occur, and some other synapses between the horizontal cells, amacrine cells, etc. Then the ganglionar layer, that is where the bodies of the ganglionic cells are, and then the optic nerve fiber layer, the axons of the ganglionic cells, and the inner limiting membrane. This inner limiting membrane is look, we have the nuclear layers. Well, external nuclear plexiform, internal nuclear, internal plexiform. Then here we have the outer limiting. Here we have the ganglionic layer. Then the optic nerve fiber. Here the outer limiting, inner limiting, the photoreceptor layer, and the posterior pigmented epithelium. So we have internal and external plexiform, internal and external nuclear, and outer and inner limiting membranes. So this pigmented epithelium of the retina is continuous with the brush membrane. When there is detachment of the retina, there will be detachment between the brush membrane, the lamina of the pigmented epithelium, that layer of the brush membrane, and the pigmented epithelium of the retina. Now, here we have the retina, and we have the peripheral retina. Well, first we have the osterata, that is here when it ends, and these inner layers of the retina will be continuous with the ciliary body. The other one, we would have, here we have the peripheral retina and the central retina. In the central retina or in the posterior retina, we have the macule. Here's the macule. So, let's see the macule. The center of the macule is the fovea. Then we have the perifovea. Then the parafovea. Inside the fovea, we have the foveola, and let's say the point of the center is the umbo. All this constitutes the macula. So the macula is a avascular part. It has a yellow discoloration in comparison to the retina, and the macule, when we have the macule, is when there is only one layer of ganglionic cells histologically. In the fovea is where the fovea, there are only cones and no rods. So on the external nuclear layer, where the photoreceptor nucleus are, we don't have the nucleus of the rods because there are no rods. So this layer would be thin. And then that is when we have the fovea. The fovea would aid on the discrimination of the vision, and the foveola would aid on the visual acuity. So now, the pigmented epithelium of the retina contains pigments that is derived from vitamin A, and it is degraded when we are seeing a luminous stimulus. When we are looking at a luminous stimulus for a long time, you see that you cannot see very well, something like that, is because there are no regeneration yet of the pigment on the pigmented epithelium of the retina, because the regeneration occurs on the dark. Now, here, this part of the retina, where it is entering the optic nerve, retinal artery, and is exiting the retinal vein, is called the papilla or optic disc, and it is on the nasal part, is the most central that we have. And remember that on the vision, what is on the upper retina is our inferior visual field, etc. So what is on the nasal retina, it is the temporal field. So because here we don't have photoreceptors, because it's this entering of the optic nerve and the blood vessels, so there are no photoreceptors. So on this nasal part we will not receive any visual information. So this corresponds to our temporal field. So because here we have the papilla on the nasal retina, this corresponds to our blind spot on the temporal side. The blind spot is on the lateral sides of each eye, not on the nasal part. So from this optic disc or the papilla, we have the retinal artery, and in the retinal artery we will see a temporal branch and a nasal branch. The temporal will be divided into superior and inferior, as well as the nasal branch, and the veins accompanies the arteries most of the times. The arteries would be a little more on the caliber. They are thinner than the veins, and the brightness is different. So when we want to see the eye, the fundus of the eye, we are looking through the pupil, this space, and we are seeing the retina, and we want to find the macula, that is kind of yellowish, but the fovea is bright red, and the optic disc, that is where the temporal arcades of the blood vessels lie. So that is, we have already seen the inner layer, vascular layer, external layer, and the vitreal cavity, and the anterior chamber, posterior chamber.